afternoon, Bob.
Good afternoon, Leighton. How you doing?
I'm doing well, and welcome back.
Well, I'm glad to be here, but I got to warn you, okay? The Carolina Hurricanes won the Stanley Cup last night, so this town's going nuts right now.
So what happened?
The Stanley Cup, National Hockey League,
Oh,
the Carolina Hurricanes. Who ever heard of playing hockey in North Carolina? Well, we won the title last night, so
well,
everybody's.
congratulations. That's
Oh
awesome.
well, yeah, I did a lot to make it happen. Just want to make that clear.
Hey, you were there cheering, right? That's all that
Yeah,
matters.
well, everybody was cheering, but it's good for North Carolina.
So yeah, a friend of mine from New York's the Knicks won,
Knicks, yeah, yeah.
and so they got smart and they parked two school buses out in the open so that the people coming out of the game could blow them up.
Could blow them up. I saw that. Welcome to New York. Okay, I live there.
The insanity of humanity, but whatever.
Right.
Hey, maybe it worked, right?
Yeah, well, something worked.
All right, so we're going to dive right in and we're going to kind of focus more on what drove you to collect that first microbe.
Yeah, well, what I'd like to cover today is let's go through some of the background, reminding people of what it was that had me do this work in the first place. Then what I'd like to do is go through some modes of action. What are these guys doing?
Oh, absolutely.
Okay, and why you just can't do anything. You got to have the right stuff. And then finally, I want to end up discussing the products that we developed.
Please.
And show you what we've been able to do and really how unique they are. Not just the fact that they're this consortium of microbes, but how they're produced, how they're used by the grower, by the farmer. So I think that's important. So anyway, back to sort of the beginning again, to rehash some of the stuff we already talked about. Basically, I came of age, went to school, graduate school, at the time that people started talking about, quote, organic agriculture. Okay, last week we talked about the Green Revolution and how Norman Borlaug say pour on the fungicides and the insecticides and the fertilizer and everything, and you'll be able to quadruple your crops. And he was right. Okay. Only problem is that by doing that, you were creating havoc within any ecosystem that you were using. And the other side of that coin, which I think is very important, and I remind people of this all the time. I live in the United States. The United States is 4.2% of the world's population. That means there's a whole hell of a lot of other people out there. And when you say buy fertilizer and buy this pesticide at $295 a jug, it's fantasy. It's fantasy. People cannot do that. Okay, not that they wouldn't want to do that necessarily, but it's simply not affordable. It's pipe dreaming. Okay, it's never going to happen. It didn't happen in 1963, and it's not happening today. And in fact, I would say that it's actually getting worse in terms of developing nations, poor nations, being able to have the resources to be able to grow their food. So I became interested in, obviously, I was a pathologist. So I'm looking at all kinds of problems, from fungi to bacteria, viruses, and realizing that a lot of these things are transmitted by insects. So I took a lot of entomology and studied a lot of it. And I said, there has to be better ways of dealing with this. Sort of, way back in my mind, I said, life has been evolving on this planet for 3 billion years. And here it is. Okay. And no one was watering it, fertilizing it, or pesticiding it. There's got to be something out there in nature that has the solutions to many of our problems. So that was the philosophy that I brought to it. And back when I was being educated, let's just use the numbers 1970 to 1980. Okay, this idea of organic farming, of natural ways of fertilizing, of controlling pests and pathogens was pretty damn new. In fact, you take a look at what we knew about soil amendments or biopesticides at that time. You found like 2 or 3 different things in the literature. Okay, it was just very scant literature. I found that mycorrhizae protect roots. That was the beginning of that knowledge. Or Bacillus subtilis seems to reduce pathogens. And of course, when I was at Syracuse, that's where the first studies on BT, Bacillus thuringiensis, was done, which is a bacterium that fights all kinds of insects, which now they've genetically modified things that the BT gene is in everything. But it was at Syracuse University where it was first discovered by an entomologist. And again, that was the infancy of this idea of, are there things in nature that we can use to be able to naturally control problems, naturally stimulate growth, naturally stimulate the roots to take up more nutrients and water. That's where it began. So I got this idea that I was going to create a library. That's what I call it. And the library was to go, I traveled for my job, my business, my professorial work all over the world. I have been in 108 different countries and studying all different kinds of things, collaborative research projects. And I grabbed, with my APHIS license, soil samples from various ecosystems wherever I went. And I was particularly interested in what we call extremophiles. Okay, what if I go up to the boreal forest on the Arctic Circle and collect roots and soil underneath these perpetually frozen forests? What if I go to the desert where things are dry? Some of the deserts that I visited had less than 1 inch of measurable precipitation per year. Okay, things are growing there. Okay, why don't I grab some of that soil? What about tropical rainforests? Spent tremendous amounts of time, all the way from the Galapagos Islands to the Amazon, Belize, Costa Rica, Peru, Brazil. Okay, and again, these are ancient forests. Okay, that are there. Question?
No. I was just waiting
Okay.
to someone that they popped.
Okay. So I collected a lot of this stuff and brought it back. And in a lab, I took 5 graduate courses in mycology and bacteriology, microbiology, and knew where to find the literature to be able to isolate these things. And I isolated, and I get all different kinds of crazy organisms in it. Some of them well known, some of them poorly known, and a few of them unknown to science. One of the samples I collected was from a bog. Okay, well, a bog is alive. Okay, it's highly acidic. Okay, there's sphagnum moss growing in it, but it's growing there. Why is it growing? There's got to be stuff in there. So I collected a library of these things, and one of the things, there's a word for that. Okay, it's called bioprospecting. Okay, and one of the things that bioprospectors learned a long time ago is you better live a long time and write a long will and testament, because it's not that easy to do. Not in terms of going to the places and collecting it, not in terms of isolating it, but in terms of finding something with really interesting biological activity. That's where the real rub comes in. Okay, let's talk about two common things that a lot of soil amendment companies use. They've been known since the 60s, okay? Bacillus subtilis, common soil bacterium. Okay, you can go to your backyard, I can go to my backyard right now and isolate this root inhabiting bacterium. Well, you go to the American Type Culture Collection and you'll find over 2,000 accessions of Bacillus subtilis. They're all Bacillus subtilis, but the best analogy I could use is let's use people. Homo sapiens. Every human being on this planet is a Homo sapien. Same genus, same species. Yet we have incredible amounts of genetic diversity. Okay? Our genes are different. They code for different things. How come Asians don't get this disease? How come Europeans are susceptible to that disease? Okay, even though we can all interbreed, that's the definition of a species, we carry much different genes, particularly when it comes to things like genes, when it comes to pathology, to diseases itself. So the same analogy is there. Okay, if I took all 2,500 accessions of Bacillus subtilis or all 1,500 accessions of Trichoderma harzianum, that's a fungus that we commonly use and include in a lot of our mixtures and everything, and I plated them out, no big deal, really easy. But then I started testing them. What do they do? What are their metabolites? What are they producing in nature? And even, we'll talk about this later on, but perhaps one of the most important things is that taking it into a petri dish is one thing, okay? What about actually when you put it in the soil? Does it survive? Does it do what the literature says it's going to do? And what I found out is that in 95% of the cases, the answer is no. Okay, so I can get a whole lot of different accessions of, quote, the same bacterium or fungus, but when I test it for antibiotic production, for enzyme production, for hormone production, okay, I more often than not fail. But once in a while, you hit a home run and you get an isolate from wherever and you test it and you go, wow, look at that. That's really doing something that's important. And that's it. It's trial and error. And a lot of modern science, we're in the world of molecular biology right now, where if you don't like something, you use a CRISPR gene and you jerk it around a little bit and bingo, it does what you want. And I'm not against that. Okay, I'm not against that. Although, we got a long way to go. There are many countries, like including the entire European Union, where you say GMO, genetically modified organism, the answer is forget about it. Okay, they don't want those things in their environment. They don't want it in their food supply. So I decided that even though I knew about molecular biology and worked with a lot of colleagues who did it, I decided I didn't want to go that way, but rather look at nature and what it provides, because of my faith in the fact that there are probably solutions to all kinds of problems out there that we haven't even discovered yet in the slightest way. So I made this collection. I would put them, once I got it in pure culture, I'd put them in test tubes. I'd put them in a refrigerator where they would hold for many years. I'd come back and transfer into new tubes. I would plate them out to make sure that they were still doing what they're meant to do, and got all of these things. So At that point in time, actually in 1995, okay, a gentleman from Perth, Western Australia approached me, literally knocked on my lab door, and this Aussie walks in, told me who he was, said he heard about me and the work that I was doing, and all of a sudden we were in business. Okay, he had been doing exactly what I was doing in Australia. Even better, okay, he had a contract from two companies, Merck and what was called Ciba-Geigy back then, a Swiss firm, which is now Syngenta, to do exactly what I just said. Go around the world collecting microorganisms, testing them for activity, and when he found something, he was under, a contract to send those samples to these companies. And he had done that for about 10 years before I met him in 1995. And you will recall from last week, 2 years later, those contracts ended when the International Convention on Biodiversity was signed. And basically, it was now and continues to be impossible to actually patent a natural organism. You can't do it. You can do GMOs, but you can't do it. And at that time, Merck and Ciba-Geigy were heavily into cancer research, okay? Cancer therapeutics. Why? Because vincristine, vinblastine were discovered right here in the Research Triangle Park by scientists at Ciba-Geigy, and they're still used today for leukemia. And these were natural products. Okay, Mother Nature's pharmacy, that there was a scientist working on it. They both come from Madagascar periwinkle, of all things, a beautiful flower that a lot of you people use in your garden. It's a gorgeous plant. Okay, but this guy was doing extracts of these things and then testing it. He was using it in live cultures of tissue samples of tumor cells. And holy cow, I mean, it's remarkable research. So these two companies got the idea that they were going to hire people to do bioprospecting, including my colleague from Australia. And it pretty much died in 1997. That's where all the big money was coming from. Last week, I talked about Shaman Pharmaceutical out in San Francisco. Same idea. This brilliant woman found a fungicide from a plant in the Amazon, isolated it, was able to produce it, and she was receiving hundreds of millions of dollars of funding, and the whole thing fell through when these funders determined, sorry, can't patent that. And I'm not going to get into the wonderful world of capitalism, okay? But these multinational corporations really do not want to spend what could be billions of dollars on something that they don't have total control over. And I get it. I'm not stupid when it comes to those things. So a lot of what I did and do really fell by the wayside. Okay, there was a period in the late 1990s, early 2000s, where ethnobotany, the famous Dr. Schultes from Harvard University, the father of ethnobotany, did exactly what I was doing. He centered his work in Panama and the Amazon. And he discovered, imagine this, 113 different plants, discovered over 1,000 different compounds, an icon. Okay, study from, this must have been nice and everything. There's a great story. At the end of World War II, he went off to war, but he came back and to his Harvard professoriate, had a wife at that time, and gave her a kiss on the cheek and said, I'm going to South America and I should be back in a year. That was the famous story that's told over and over again. 13 years later, he came out of the Amazon jungle with his samples that were then sent back for taxonomic work and chemical work. And he literally blew the socks off of the world in what he did. There aren't a lot of universities who let you take a 13-year sabbatic without ever communicating with anybody, but that's another story. So basically, we're in a situation where I took a lot of interest in, another word for this thing is natural products. Okay, these metabolites that come out of these microorganisms that I use are natural products. Okay, produced naturally by the organism. And we look at what these things can do. And either for better or for worse, we all know about the fate of the Amazon. It's estimated that we have discovered less than 5% of the organisms in the Amazon. And of those 5%, we have screened less than 1% of them for actual biological activity. So Lord only knows what is out there waiting to be discovered. As I said before, once the whole idea of not being able to patent these things became off the table, a lot of interest went out of it, because to do research, it takes money. So I was fortunate enough to get enough grants and to do my work. Never got rich from doing it, but nevertheless, I carried it on. So this guy from Australia, we started a company called Natural Science Center. He had the company was based in Alabama, and he had these giant bioreactors where he would put his cultures with his, he used mostly a molasses yeast mixture to be able to grow them and produce a number of bottled products. Okay. One of them was called SC27, which stood for Soil Conditioner 27. Okay, that's all it meant. It had 27 different organisms in it. And he tried real hard to establish a company. And for a few years, we did okay, but it would never gain a traction due to the fact that this was back in the days where a lot of biologicals were simply called snake oil. Okay, people didn't believe it. And of course, the ag chem companies certainly didn't do anything to promote it. I think that is where most of our problems came from. Tragically, he passed away suddenly in 2011. And we were left sort of holding the ball, and things went south very quickly because of financing. And one of the real problems, and we'll talk about this in a little while, is that we sold these products in 2.5-gallon jugs. Okay. And that 2.5-gallon jug was enough to treat about 1 acre of whatever you were using it on. 2.5 gallons to ship. The cost of a plastic jug, the label, the storage, the packaging, and everything literally is enough to put you out of business. Okay. The cost of simply being able to do it, is twice, 3 times, 4 times as much as actually creating the product in the first place. Not the R&D, but the actual creation of the product. Okay. A quick story that I think is worth talking about is that one of our concoctions was something that worked. These are, again, natural organisms, fungi, bacteria, that worked incredibly well on nematodes. How'd we do that? We did the experiments. We gave away a lot of material where people had problems, and they went away. And they created a product called SC27-N. Okay. And we were selling some of that particular product. Couldn't be terribly cheap. Again, the cost of just getting it from one place to another was absolutely ridiculous. One day I was not there, but our lab technician was at the lab, and there was a knock on the door, and a guy with a black suit is standing there and said, hello, Mr. So-and-so from the United States Environmental Protection Agency. Is this Natural Science Center? And he said, yes. And he said, can I have a minute of your time? And they said, sure, come on in. It's our understanding that you're selling a product, SC27N, that's for nematodes. And they said, yes, we are. And basically, without going through the details, because it's irrelevant, we were told that we had 30 days to pull the product off the market and to destroy any of what we had stored and get rid of any advertising on it, because by saying that it was a nematicide, we were violating federal FIFRA law, Fungicide, Insecticide, Rodenticide Act of 1972, okay? Where in order to have something with the word side on it, you had to have an EPA registration. Okay, we were selling it as a soil amendment, but they didn't want to interpret that way. But it simply shows you some of the problems that you encounter trying to do natural organic work. So without going again through a lot of the stories, I met a bunch of gentlemen out of Atlanta, Georgia. They knew about what I was doing and knew about my collection, and said, do you think you could create another product, because those other companies are done? Okay, I said, well, I could certainly try. I thought I was out of the business. I haven't thought about it, but I certainly have those cultures. And lo and behold, okay, within 6 months, we had a preliminary liquid product that we named Rhizol. Okay, I'll take credit for that. In Latin, rhiz, R-H-I-Z, is root. And where did I get all from? I said, well, all the drugs out there are like Tylenol, okay? O-L. I said, we're going to put Rhizol, okay, on it. And I made up a word, and bingo, we had a product. So we were smart at this point, okay? First of all, we started doing massive amounts of different trials. And I'll go back to the mode of actions in a second. But I'm getting ahead of myself. But the bottom line is we looked around at other competitors in the soil amendment business and found out several things. Number one, what I call the same old, same old. Okay, 3, 4, 5 different microorganisms that have been known for a long time. Even some of the big corporations like Bayer Crop Science was selling Serenade. What was Serenade? Bacillus subtilis. They got an EPA label for it. One microbe. Okay. It costs over $500,000 to get an EPA registration. We didn't have that kind of money. Okay. We take a look at Novozymes. They still have a product called Actinovate. Okay, Streptomyces liticus, well known in the literature. They got a license for it, they registered it, and it was done. But the one thing we saw was the same problem that we encountered back in 1995, is you create a jug of this stuff. You got a couple of problems. The first problem is that many times it has to be refrigerated or it dies. Other times it has a shelf life on it. Okay, you got to use it within 6 months or throw it out. Okay. Additionally, you've got the entire problem with distribution, that you have to have gallons of this material to be able to treat a relatively small area of ground, whether it's a garden or a commercial grow. So we said, we got to do something about that. So I'll give you a hint about what I'll talk about. I'm going to put up this bag. Okay. Okay. This is a bag of Rysol. Okay. And this one's for cotton. Okay. If you take a look at this bag, it's 11 inches this way. It's 6 inches this way. And it's less than 1/2 inch this way. This contains enough of our product to treat 40 acres. There you are. Weighs less than a pound. Okay. 40 acres. Well, that's a big deal. That's a real big deal because now you could ship a crate of this stuff for the price of a box of books. Okay. And I'll talk about the actual product and how long it lasts and how it was put together later. But that was the idea. Okay. I'm working with some really smart people, both in business, but we also have an engineer that said, yeah, I think I can do that. And my response was, well, no one else could do that. Okay. And we had to buy some really expensive freeze dryer material equipment, which he had to completely modify. Okay. Again, patent, okay, protected, okay, the modifications to create these products. And I'll give you a teaser. Okay. In a little while, we'll talk about these. That was a bag. This is a pill. Okay. You toss one of those pills in 5 gallons of water, and bingo, you've got a product. So, what we had to do was do something that was different, that not only was able to meet the needs of a grower, but was able to actually be able to be effective and effective over a long period of time without refrigerating it, without freezing it. Okay, quick story. I'll never forget. Back in the SC27 days, again, the jug of brown liquid, we gave a whole lot of it away to some grower in Louisiana. And he basically, we're giving it to you for free. This is how you apply it, yada, yada, yada. Well, again, long story made short, the guy left the jug out in his field in the sun for a week, okay, before he even opened it up to put in his sprayer. Well, basically, I would imagine that the temperature inside that jug probably got raised to 125, 135 degrees Fahrenheit. He basically cooked everything. Okay. So again, these are the limitations of using things like liquids. We'll get back to that. All right. So the next step, I got all these bugs, we call them, okay, microbes. And I said that I have to do a tremendous amount of lab work to screen them for activity. And the 3 major things that we were looking for, I mentioned them before, is first of all, antibiotic activity. Okay. If I take this fungus and I put it in this bacterium, what happens to the bacterium? Okay. If I take this bacterium and put it in with a bunch of nematodes, what happens to the nematodes? So that is done, as you well know, in vitro to begin with. That's in beakers, in flasks, and on plates of different selective agar to test for actual activity. And that's, again, the screening that takes immense amounts of time, okay, to be able to screen these things and find out, is it a plus or is it a minus? Are we getting an effect or are we not getting an effect? The second is enzymatic activity. Okay, one of the most important things that microbes do, pretty much all microbes that are saprophytic. Saprophytic means it lives on organic matter. Okay, soil has organic matter. You till your corn stover into the ground, it's filled with organic matter. You come back next season and it's gone. Well, where'd it go? Okay. And where it went is that it was eaten. Okay, saprophytic microorganisms used that dead plant material as food. The major source of food for microorganisms is carbon. That's the backbone. We are all carbon organisms, carbon-based organisms. As anybody out there knows, take organic chemistry. You'll learn all about it and you'll get a headache, okay? But the whole point is that how good are the organisms that I'm using in being able to first digest organic matter? How efficient is it at doing that? Secondly, what does it do to that organic matter? Okay. Does it transform it? Okay. If I take a leaf and put it next to a root, the root can't do anything. Okay. The root needs nutrients, it needs nitrogen, potassium, phosphorus, 21 micronutrients. Okay. It can't use a leaf, even though the leaf may contain all that stuff. So it's the microorganisms that digest it and through secondary metabolism transform that organic matter into, a good word is soluble. Very important because plant roots can only take up water-soluble nutrients and make it in a form that's available to the root itself. The most notable of all these organisms are rhizobacters. Okay? Basically, the air you're breathing right now is 71% nitrogen. One of the most important nutrients to a plant is nitrogen. You don't have enough of it, you ain't got no plant. Okay? The three biggies: nitrogen, potassium, phosphorus. Well, obviously, nitrogen shouldn't be a problem if 71% of the entire atmosphere is nitrogen. Well, the good Lord played a trick on us, okay? Nitrogen in the form of N2, that's what I'm waving around right now, is not available to plants. It can't do a damn thing with it, as in nothing, zero. It has to be in a form of being nitrate, nitrite, ammonia, urea, okay? The breakdown products of organic nitrogen, which takes place in the soil on the root. So, I mean, this was discovered, one of the most famous, okay, is soybeans. We have soybean inoculum, okay? Rhizobium inoculum. It puts little nodules on the soybean roots, and those nodules actually take the air that's in the soil. Well, you people better know that the soil has air, same air you're breathing right now, and converts it into an available form of nitrogen. Hence, nitrogen in plants, legumes like soybeans, make their own nitrogen. It's self-feeding. Okay, and that's why it became, something, a very important protein crop, is that here's something that you don't have to add nitrogen fertilizer to. Okay, you want to talk about nitrogen fertilizer? Most of the nitrogen fertilizer in the world, chemical fertilizer, comes through where? The Gulf of Hormuz. And it stopped. Okay, all around the Gulf, there are corporations that take natural gas. They got lots of that, using the Haber-Bosch transformation, Nobel Prize winning work, German scientists, 1926, is able to convert natural gas into ammonium nitrate fertilizer. Granule, put it in a bag, put it on a ship, take it all over the world. Okay, that's where it comes from. So when it comes to non-legume crops, we got a real problem, okay, right now. And if you're a farmer, God knows you know that. Okay, people are offing themselves due to what's going on right now. but legumes do it naturally. So that's just giving one example how rhizobacters are able to create the own nitrogen. But what about potassium phosphorus? Okay, what we have is that these organisms take that organic matter and even take the mineral soil, and through enzymes, okay, maybe some of you don't know what an enzyme is. When you eat your food, okay, your body can't do nothing with food. It goes into your stomach, and there's a whole bunch of enzymes in there that grind it up and break it down into amino acids and glucose and all the things that are being done. Same damn thing in the soil. The soil is a couple of 100 billion acres of stomach, okay? And the microbes that are in it break down, in this case, not only the organic material, but the inorganic material, okay? Those grains of sand, grains of clay, grains of silt, called mineralization, demineralization, actually breaks it down and makes these elements soluble. Back to that word. They can be solubilized into the soil water that comes in, the roots take it up, bingo, agriculture. That was number two. The third is perhaps the most interesting thing that I'm looking for, and those are hormones. Okay, you got hormones. That's what makes your body function, your pituitary, your thyroid, your adrenals, your gonads, critical organisms, critical organs that allow your body to function. Without those hormones, you're in big trouble. Okay, if you got thyroid problems, they give you a thyroid pill for the rest of your life every day in order for you to live. So in the soil, where hormones react is in plant growth. Plant growth. And do you think to a farmer that's important? Well, it's real important. Okay? Plant growth does what makes the plant go through its physiological stages. Looking something up here just to show you something. I'll put this other phone up, okay, because I was looking at them this afternoon when I got back from the lab. Those are some tomato plants that I grew from seed, okay? I put the little tiny plants in the ground on April 30th. I've harvested 10 pounds of tomatoes so far from those plants. And those plants that I just showed you are 7 feet high. That's a tomato I just took this morning. I'm going to tell you what that is. This is June, the middle of June. You're not supposed to be able to grow tomatoes from April 30th with a plant this big to a plant 7 foot high with mature tomatoes on it by June 15th. You'd expect it more like August 15th. Those plants received one of those pills that I'll show you in 5 gallons of water. Drench the soil when I put it in. No fertilizer. Zero fertilizer. Just natural soil that I've got in my yard. Not saying there weren't nutrients in there. Obviously, there were nutrients. But what is the point? Just showing you that little silly anecdotal experiment is that hormones are what? First of all, make the plant grow a robust root and a robust shoot. Different plant hormones code for different things. We have gibberellins, we have naphthalenic acid, okay? We have all different types of plant hormones that were discovered about 50 years ago, okay? And the reality is that what we have discovered is that hormones are not only naturally made in the plant at very small levels, but are produced, back to that word, as secondary metabolites in microbes. certain microbes, quote unquote, poop them out as part of their eating process, a part of their life cycle. Those hormones are very water soluble. The hormones are taken into the root. Even remember we talked last time about vesicular arbuscular mycorrhizae, endomycorrhizae. Those are fungi living in the root. And we thought they were pathogens up until about 25 years ago. Okay, well, they're not pathogens. They're actually natural organisms that not only help the plant in a lot of different ways, but they produce hormones that incite root growth. Okay, mycorrhizal, ectomycorrhizal fungi. Remember our root, okay, and that big fungal sheath that goes around it, okay? The root gives it some glucose. The fungal sheath gives it everything else it needs, antibiotics, enzymes, and indeed hormones. You take a look at a plant that's inoculated well with ectomycorrhizal fungi, the root system is 3-4 times the size because the fungus elicited a hormonal response in the roots. Goes further, okay? Let's talk about cannabis for a second. The whole big deal with cannabis is the flower, okay? You're looking for flowering, okay? You're looking for a big cola filled with buds and oil. That's the business side of the plant. Most of the rest of it is garbage unless you're making hemp products. So what's the point? We're learning, and we're still in the early stages of this, folks. The amount of physiology being done with cannabis and a lot of other crops is astounding. What we're finding is that the timing of the set of those flowers, the development of the flower, the size of the flower, and the amount of oil that's deposited in the flower is all hormonally regulated. Therefore, by having the right combination of microbes in the soil, producing those hormones, telling the plant, oh, it's time to do this, it's time to do that. Okay. How about a treatment right at flowering? Give it a boost. We found that worked significantly. Okay. You get this hormonal relationship, in this case, not so much with the root, but with the shoot and with the flower. Okay. We did a huge study. This is one of the first studies we did. We got some good funding back in my 1995 days from a private corporation who believed in us. And we spent $200,000 doing horticulture studies. Horticulture studies. What is horticulture? A lot of people don't get that word. That's all the stuff you go and get at the garden store, all the pretty plants, okay? All of the shrubs.
Uh-oh.
Okay, I think he tried to take a phone call again, or is trying to stop a phone call. We'll see if he pops back on. We'll see if, oh no, he had to jump right off. He'll come back in a second. But this is some amazing information, Leighton.
Yeah, just to comment on Silly's comment about me not speaking. I didn't want to interrupt him because he was on a roll, right?
Yes.
I was allowing Bob. Thanks for coming back on. We're just talking about. Silly made a comment about the fact that this is the first time. I literally haven't spoken for 49 minutes, sir.
Is that a record?
You have the record, sir. By far. Yep. By
far. Okay.
The reason why I didn't stop you was because you were creating a...
Okay, I can't hear you now.
Uh-oh.
Hello?
We can hear you.
Can you hear me, Bob?
I can hear you.
Okay, now we can't hear. Leighton, jump off, jump back on. We'll try to get...
I can't hear Leighton again. Can't hear Leighton.
Yeah. Well, that happened on the last show.
That happened to me last time, I know.
Yeah, so I'm not sure what's going on. Every time you get a call, it seems to want to take one.
Okay, now I can't hear either one of you.
Oh, jeez. Okay, I'm gonna kick you
It
out.
says
come
you're
back,
in the
Bob.
show. Everyone can see you and hear you. What can I tell you?
Okay, Leighton,
All right.
speak. Let's see if he can hear you now.
Can you hear me now?
Cannot hear Leighton. I can hear you.
That's got to be on his end because the opposite happened last time.
It's very
So-
strange.
So, Ken, please tell
Very
us.
strange. Cannot hear
The
Leighton.
reason I did not stop his conversation was because he was weaving.
Do you want all of us to just go off and jump back in again on the page?
Tell
Let's
him
do
to-
that. Okay, let's do that.
Okay, guys, I think our audience can hear us, but let's see if we can get things working right so that everybody can hear everybody.
So anyway, back to what I was saying.
Yeah.
I just didn't feel it was important to interrupt it. I liked the way he was weaving. I mean, this is all the same shit we've been talking about for years on this podcast.
All right. Well,
Can you
we're
hear me?
back. We're back, and I can hear you fine. Correct.
Okay, can you hear me as well?
I can hear you as well. I got a message that the CIA has interrupted our broadcast. Okay.
Clearly.
Okay,
But,
they don't like this at all. Okay.
yeah.
So if it
that's-
happens again, Bob, remember to jump off and jump back on. But I'm going to
That's
jump
it.
off and let Leighton talk for a second.
Okay.
let
Talk,
me-
Leighton.
For the first time today,
Now I can't hear Leighton.
you can't hear me?
Okay, I have no idea. Can you hear me,
Now
Bob?
I can't hear either one of you.
Jump off.
Okay, let's try it again. Off and on.
Interesting. But anyway, yeah, so it's not like we don't talk about that. It's not that the lesson hasn't been taught 150 times over the last 8 years. But I liked the way he was weaving it together in a big picture. He
Yeah.
got a little long-winded on some of it. I love the little stories and the anecdotes. That was all perfect. I would have boiled him down a little, but by interrupting him, wouldn't have gotten that same woven.
Hello.
Can you hear us now?
I hear you now.
Okay. I'm not going to jump off. I'm just going to stop my camera because
Okay.
it seems that's affecting things. So I'll stop my mic and camera and you guys can carry on.
Sounds good.
All right. So you've woven the path. You've done a brilliant job.
Right.
You got to the end where you talked about the three principal things that you wanted to achieve in
Correct.
your you've demonstrated completely 100% on the importance of each one of those three goals.
Yes.
And now let's get into how did you figure out that maybe the 27 were the right ones, or 40?
Okay. Good question. Let's move on to what we did. Okay. So we're up to the point where a couple of things, okay, came about in this new company, which is called ProGro Bio. Okay. ProGro Bio, we sit down saying, what do we need to do to really change this industry? And I said, we've got to have a consortium of microbes that A, will live together, very important. B, will live in the soil, not just on a petri dish, very important, okay? And is incredibly easy to use, okay? It doesn't matter where you store it, doesn't matter what you do with it, okay? You put it in a bucket of water and you could use it in a tractor, an irrigation system, a drip irrigation system, in a watering can, in a backpack sprayer, and it's going to be there. And we'll get into that when we talk about the products. So we're up to the point where I've got all these organisms, hundreds and hundreds of organisms, and I asked the question that we just posed. What of these organisms can do those three things that I call biological activity? And the answer is, as I said, you begin by working in the lab. You get your candidates. Then I was very fortunate to have a greenhouse to work in, okay? And you get your plants potted up. and you put it in with a sterile soil. Okay, so there's no crossover, and you inoculate that soil with what you got, and then you start looking at what's happening. Okay, what's happening in the soil? Are they multiplying? Are they getting onto the root and into the root the way they're supposed to? What about the controls that are not inoculated? What about the root biomass, the shoot biomass, the flower biomass? Oh, I just remembered what we dropped talking about. Horticulture, very fast, okay? Because we were talking about flowering. We had 18 species of flowering bushes and plants, from roses to carnations to hydrangeas to dogwood trees, okay, that we tested. And again, make a long story short, this was done in 1996, 1997. You want to talk about a long time ago? We found that the flower output, which is why you're buying these plants, was again triple to quadruple as compared to the controls, which received normal fertilization, normal water. So that's just another example.
All right. So you're going. All right, let's back it up. So
Okay.
basically, the information that you're providing, we all know that's why you're on this podcast. You're dealing with a bunch of guys that are really intelligent. They love this stuff. They love the meat and potatoes. I purposely didn't stop you because you wove a beautiful story together. But now let's get into some meat and potato stuff, right?
Good.
So we know about all of the benefits of microorganisms. I personally have got products that do something similar.
Right.
But I have never seen anybody pull 40 microbes from all over the world, figure out what they did and how best to blend them. And that's where I'd love you to talk a little more.
Okay. Great question.
Are each microbe equal, the same number
Okay.
of
Great question.
There's some that you have a lot, like 50% of them is this one organism, and the other 50% is made up of different pockets. So talk a little bit more about the science.
Super question.
Yeah, that you did to create this.
Okay. The science behind it. Okay. So we're building a timeline here, guys. Okay. We now have these microbes isolated. Okay. We have tested them in the laboratory, in the greenhouse, and in field trials. And we said that there's something really important going on here. The first question that you should be asking, and we were told, by the way, it can't be done. I want to make that clearer. What I'm about to say can't be done. You build a manufacturing laboratory. You build bioreactors. These are stainless steel tanks that you could do all kinds of things with. Stir it, modify the pH, modify the temperatures, very critical, okay? And I want to grow these in the same frigging tank. Can't be done. Okay? Fungi, bacteria, actinomycetes, rhizobacters, and everything. Well, we're back again to trial and error. It began in my laboratory here in the Research Triangle in containers, Nalgene containers, okay? And we went through minimum of 40 different nutrient combinations. That includes the carbon source, vitamin sources, everything, pH adjustment, different iterations, some from the literature, some just coming out of our ears, made it up, okay? And what did we find out? That most of these didn't do very well, okay, when you're trying to grow 40 organisms at the same time. I know how to grow Bacillus subtilis. I know how to grow Streptomyces pseudomonas, okay? Can I put them in the same tank and be able to grow them? Well, lo and behold, we came up with a final liquid solution, nutrient source that worked, but it didn't work commercially yet. So now we take that chemistry, we bring it to the manufacturing lab. We take a 40-gallon bioreactor, we fill it up with that nutrients, and we inoculate it with all the organisms. Well, you ain't there yet. Okay. Some of these organisms like higher temperatures, some like lower temperatures, some like a pH of 7.8, some like a pH of 5.1. Well, let's give credit to our engineer. And again, this is not some sort of a bing, bang, boom, there it is, we did it. Trial and error over and over and over again in order to create the environment and the nutrient source in which we were able to brew a incredibly concentrated liquid material. Okay, it starts off almost clear. Now it's this thick, almost goop that contains these. Yes, go ahead.
That's meat and potatoes. That's what I'm talking about. All right.
That's meat and potatoes, but we're not there yet.
Created an amniotic fluid.
Yep.
The perfect liquid environment for to stimulate all of the organisms.
Yep.
Then you figured out how to adjust the pH in a way that everybody was happy.
Optimal, correct.
Yep. And then you also figured a way how to deal with the temperature.
Yes.
Everybody was happy.
Yes.
All right. So you jumped through 3 major hurdles. What happened next?
Okay. Now, bingo. We got a product. We took that liquid and we put it into trials. And we were getting some unbelievable results, highly significant results. So now we're back, okay, in the days of, all right, I got this jug of muck, okay, we could put it into a plastic container. And by the way, that's problematic because you tell me, Leighton, if I take a bunch of living organisms and I with the nutrients and throw them into a plastic container and shut the top, what's going to happen?
They'll consume all the oxygen and it'll go anaerobic and crash.
It's going to go anaerobic and die, but what's going to happen probably before that?
Probably explode due to the hydrogen.
Boom! The jug is going to literally blow up. Okay.
I think you heard.
So you got a problem here.
I think you heard.
Whoops, Leighton, I can't hear you now.
Oh, Jesus.
Here we go again.
Oh, man.
Okay, I hear you now. I got you.
Okay, all right, cool.
Okay.
Nope, I don't. I got Ken.
You can't hear me?
No.
I got neither one of you.
Okay. Can you hear me, Bob?
Okay. Yep. Cut it off again. We'll go on.
So let's tell him to lose his picture.
Okay.
Because maybe that's the problem, is he's using up too much bandwidth.
Yeah, it could be. I'm going to kick him out of the studio and let him come back on again. Yeah, I don't know what's going on. Maybe it's because he's using a cell phone instead of a computer.
Yeah, it's bizarre.
Here we are. I'm back.
All right. Okay. Cut your camera. Take your camera out of picture.
My camera?
Yeah, because I think that's what's causing the problem.
You know what I'm going to do? Let me move to a place that might have better connection.
Do you see how many bars you have at the top?
Yeah, I've got 3 bars, and it says.
Find 4 bars.
Okay, okay, it just jumped up a bar, so I think I'm good. Okay, let's continue.
So we got talked about that last week when I blew up that lab in Colorado.
Correct.
Of what happens in these environments. So
correct.
How did you overcome that phase?
Okay, so we went back to our original business model. Okay, we've gone through a whole bunch of steps. We found some incredibly bioactive materials. We are able to produce it, okay, make them grow like crazy, multiply by billions X. From that, we had juice. And we said, wow, how do you make a business out of this? So the idea came up. Lyophilization, big word, called freeze drying. Okay? Now, freeze drying is as much of an art as it is a science. Okay? You just cannot freeze dry anything. And in fact, one of our major ingredients, which is proprietary and I can't talk about it, the literature says it can't be done. You cannot freeze dry this material. Hello. And I told that to our engineer, and he said, yeah, watch me. Okay. And we bought him $500,000 worth of very nice commercial top of the line lyophilizers. And he got his tools out. And apparently, temperature, pressure, valve torque, I mean, way above my pay grade. Okay. There's all kinds of things that go into freeze drying material that are, that are flowing there. You got to make it right. Okay. Remember, you're freeze drying something. You're taking something to below 100, minus 100 C. Okay. Okay. And that's what these machines do. But the question we had, okay, initially when we first started our runs was, Jesus, we're going to kill everything, okay? We're taking this wonderful microbial soup and we're putting it on the pans and we're turning on the electricity and we're going to get something, okay? Maybe it's good for milkshakes or something, but it sure ain't going to work as a microbial inoculant. Trial and error, trial and error, trial and error. Bingo, we end up with, as I said, this. Okay? This bag, less than a pound. I gave you the statistics on the size and its thickness, less than half an inch. That is good for 40 acres of treatment. Now, let's go into the mechanics of this thing. All right, we got this powder that has now been lyophilized. We tested it, and it is super concentrated. There are other powdered microbial products out there. You've got to add 6 zeros minimum per gram of the concentration of microbes for each one of our organisms that are found in that bag that I just showed you. Sometimes it's well over 100 million times more concentrated. So, wow, we came up with something. Well, okay, whoa, time out. Okay, how do you use this crap? Okay, well, again, due to the better living through chemistry, I guess. Everything I just showed you in there, if I took that bag that I just showed you, Mylar, zipped it open, okay, hermetically sealed. When it's shut, it's shut. Ripped it open and poured all that stuff into a 500-gallon tank of water. Within 5 minutes, it will be completely dissolved and suspended in that water. When I show you my pills, that takes less than 50 seconds, okay, to go and you could put it in. Yeah, go ahead.
So when you dehydrated it, you dehydrated the food as well.
That's correct.
When the food rehydrates,
it starts feeding the microbes immediately.
Have you changed it?
No.
Okay, so you literally just took the water out. You didn't have any kind of chemical reaction.
Nope. No chemical reaction. Took the water out. Okay. There are various solvents that are used in the freezing mechanism, which are not in the product itself. The product is 100% organic. There's nothing different about what is in that bag as to what was in that bioreactor tank.
So you used a solvent to make those ingredients dehydratable.
That's correct.
Wow. Damn.
Yeah.
Okay.
Okay. Again, this is all patent pending stuff because, again, no, I'm not trying to hide anything. All I'm saying is that I don't know how many times we were told point blank, you can't do that.
Oh, believe me, I've heard it a million times. That's what keeps me going, my friend.
Okay, you can't do it.
I go.
And I take no credit for that. Okay, we've got some great people who know what they're doing. Okay.
Okay.
So now we have this stuff that becomes instantly soluble.
Yep.
Okay. Remarkably, let me say something that I think is important. And I told you last week, I do not mention other products. I have tested all of them, 100% of them. If something new comes out, my credit card comes out. Okay. I bring it back to the lab. One of the first things that I look for, because they say it right on their label, is water soluble. No, it's not. No, it's not. They use clay. They use various carriers in their powder. And you take a beaker and you pour it in and you don't even have to swirl it around with this, but you can swirl it and swirl it and swirl it. And you got all this great crap at the bottom. Mm-hmm. So the first thing I discovered is no, it's not totally water soluble. This thing, you drop some in water, come back in 5 minutes, it looks like you got dirty water. That's the end of it.
That's what I wanted to ask.
Yes.
Does your product have any turbidity whatsoever?
Very slight.
Okay, so
very slight.
It's like clear water.
It's like clear water, and this is very important too. A lot of these products, okay, that claim to be completely soluble that are not. One of the funnest ways, I love doing this, I've done God knows how many experiments, is put Rhizol in a feeder tank and send it through drip irrigation, okay? Now, you want to screw up drip irrigation really fast. Use anything that has any type of grit or non-soluble material in it. You'll clog up the lines and you'll probably end up with a lawsuit, okay? This stuff is completely soluble to the point where any form of application is fine. I'm going to say something right now since we're on the topic. Let me hold this up. Remember I said we sat around the table thinking, how do we create a product that's going to answer all these questions? Well, we first came up with, liquid's great, powder's great so long as it's completely soluble and can go in any mode of application. Well, we found out that a lot of people use talc. Okay, they coat seeds or they have a talc dropper that as it's planting plants, it drops a bit of talc in the ground next to the root of the plant that usually contains fungicides, insecticides, various things. And a lot of people use this technology. Cotton in particular uses this technology. And we said, well, what the heck? We'll take some Rhizol powder, we'll mix it with talc, we'll put it in a bag and see if it works. And lo and behold, it works. So, the way we advertise it, and should advertise it more, is simply the fact that any modality that you are using, from a watering can in your garden, okay, a lot of people, a lot of moms, dads, kids got watering cans in their garden, up to a guy who's growing 10,000 acres of corn. There is a way of applying this product to that particular grow. And
I think what you're trying to say is that your products can be used in every existing methodology of delivering.
That's it.
Whether you're using it in a seeder, whether you're using a pivot, whether you drip irrigation.
Pivots work great. Yep. So,
You know,
so here, back to the utility again, okay? I got this bag. This is enough for 40 acres, okay? I sell a farmer growing 400 acres solid of soybeans. Okay, he buys 10 of these bags. He's inoculated his field. Bingo.
all right.
Okay.
This
How much liquid would I have to use?
oh, forget it.
Basically, how many barrels, note the word barrels, of other products powder would I have to use to put on 400 acres? It's not
Ridiculous.
tenable.
Yeah, ridiculous. All right.
Yeah, it's not tenable.
Hang on. So, shit. What was it? It was really important.
You were talking about it could be used in any modality. Yeah.
No, inoculant.
Okay.
The word is inoculant. Most
Yes.
people say this is an inoculant.
Yes.
And you have to use it again.
Nope.
And more often than not, you have to increase the volumes that you're using.
You got it.
Are your organisms present forever, or do
Okay.
you come back?
That is a fantastic question. Okay. There's several answers to that. Okay. As you pointed out, a lot of the competitive inoculant amendment products out there. I mean, some of them say use once a week, which
Right.
just sort of blows my mind. Okay. Use once a week. Right. Okay. A potato crop is in the ground for 5 months. I'm going to use it once a week. Okay. Those potatoes must be made out of gold. So the point is that we have two recommendations and they differ depending the crop you're on. One is at planting. Then optionally, but we encourage it at the critical flowering time of the plant. For example, with potatoes, great crop. This stuff works incredible on potatoes. Okay, you stick a potato, stick the potato piece in the ground, sends out its shoots. Okay, it starts growing a nice canopy of leaf. Okay, it's not until the flower starts that you start getting tubers set in the ground. We say give it a booster at that time. And we did the experiments to show it, and you end up increasing the yield of potatoes by 30, 40, 50% by doing it. Cannabis, same deal. At planting, put it in, and I'll explain something in a second. At flowering, the critical point where that plant has to do its business, give it a booster shot. So here's an answer to your question. Depending upon the environment, okay, all our microbes need are carbon, water, and a reasonable temperature. And you wouldn't believe how high that temperature could go. And our bugs still live in the soil. I'll tell you right now, I've taken soil temperatures of 125 F and been able to isolate all of our microbes in it. What is it doing? When it's around that root, when it has food and water, it's multiplying. One of the first experiments I did is actually on a hemp crop. And the plants were inoculated at planting. And I knew the exact concentration precisely of what I was putting in that water around that plant. I came back 3 weeks later with my shovel. Shovel now. I'm not mining microbes, I'm mining soil. I took the soil out of the ground, brought it back to the lab, extracted the microbes from it, and the multiplication factor had gone up by 1 trillion colony forming units per gram of soil. They multiply in the soil. They continue to grow by themselves. Okay? So that
they overwinter.
aha. Boy, it's smart guy here. Okay. Now we have. I told you there are different modalities. Okay. and I'm probably giving away some secrets here, okay? If I'm using Rhizol on a golf green in Tampa, Florida, okay, it don't freeze in Tampa, Florida, okay? And it is, so long as there's water and there's carbon, it is going to keep on reproducing itself and doing its thing. If I am growing sugar beets in North Dakota, the ground is going to freeze 3 feet down. Interestingly, does it kill all of our microbes, which is what I would suspect it would do? The answer is no. Most of those 40 microbes survive, but many of them don't. And the actual titer, the population goes down when you have a deep freezing event. So the environment that you are using it in is going to play a role of how long you are going to get activity once you've inoculated that particular soil or crop.
So you did answer. You are an inoculant in the right environment.
That's correct.
You're more of a product in a hostile environment, which is fine.
That's correct.
At the end of the day, it doesn't matter. The farmers are going to want they're treating their field anyway. So it's not in the world. They'll spend the money, especially like if the price goes down because they don't need as much. It's still a win-win for you. Because the reason I ask this is because I'm an anti-business. When I'm done, I'm done. I treat you 3 times and goodbye. Hopefully you give me a recommendation. Otherwise, I will not survive. Right?
Correct.
That's how I've stayed in business as long as I have. Because truly, once I've established my biota, as long as you don't use synthetics, as long as you're careful with the water quality that you use, you'll be okay. Now, typically, I tell people if you're on a well, you're fine as long as it isn't contaminated. If you're using city water, then you're going to probably have to continue to use some amount of this product indefinitely, or you get one of those water purifiers that cleans the chlorine.
Well, we've got a new pill that's coming out, okay, that actually clears the chlorine from your tank full of municipal water.
Yeah, and again, chlorine's easy to tie up. Humic acid, right? But again, there's fluoride. There's other contaminants, pharmaceuticals. There's all kinds of.
Let's answer a question.
So
you're asking all the questions that we asked. Okay, will water, municipal water from here, from there. As I told you, we did trials in 28 states. Okay, some of those trials were using well water. Some of them were using municipal water. Okay, and I had samples sent back. I said, I want to know.
Yeah.
I happen to live in an area where our water sucks, okay? They put so much chlorine in our water. If you take a bathtub, you smell like, I don't want to talk about it, okay?
Yep.
And so I said, our water is filled with chlorine. And I went and put it in a beaker and put it in our microbes. Did it do anything? No. Now let's move on to something else. Very important, okay? You just said something really critical a minute ago. We want a farmer to be able to use this material in any modality that they wish. On the other hand, my friend, as you well know, a lot of farmers are not going to be purely organic. They are going to use fungicides, nematicides, insecticides, okay, because that's what they're addicted to. And again, what's a question that we ask? Well, what if we mix Rhizol in with these other chemicals? Is it just going to kill everything? I tested 9. I asked the farmers, what do you use? First of all, I don't make it up. I got samples of those things. I did the experiments at the appropriate levels of concentration, except for one fungicide, which acts on actinomycetes very negatively. You can tank mix our microbes with these chemicals, and still spray it, because a lot of farmers say, if I have to go over my field one extra time, I ain't doing it. Okay?
Yep.
That's reality. I'm trying to deal with reality. So,
did your microbes break down or
no,
destabilize or soften the effects?
that's a really good question. Those are physiology experiments, and I've not done that.
All right, cool. Because
but
that's what you've done. You've altered the chemistry slightly enough through your microbial consortium,
yes,
to prevent them from dying, because they are from extreme environments.
Yes.
Again, kudos to you for doing it.
Yeah. I just wanted to mention that because it's important. Because again, I mean, the thing that we obsess on more than anything else is, okay, you've got 10 million farmers in the United States and 100 million in Europe, and they all do things a different way.
Yeah, their way.
And we know damn well that we're going to get questions. Can I do this? Can I do that? Can we do that? So we try to anticipate all of these modalities and then run the experiments to see if we're good or bad. And we'll tell them the truth.
All right.
Okay. Yeah.
But listen, I've got to hurry you up. We've got more shit to do.
I got products, man.
Right. Hang on. We got 8 questions, and I got one last question that I'm going to ask you.
Yes.
How the hell did you get trike and mycorrhizae to play nice together?
They play wonderfully together.
In all of my research, all of my work,
yeah,
they're always competing for infection sites. And if you use trike too early, the mycorrhizae can't get to the infection sites. The trikes are way faster at colonizing.
The best answer I could give you is I did a huge experiment for 3 years at the North Carolina State Forest Nursery. And I was using our product, which has a lot of trike and has 4 ectomycorrhizal fungi in it. And bottom line is, we were able to prevent fusarium, verticillium, pythium, and mycorrhization of roots. Ectomycorrhization of roots was 98% of short roots versus 5% of controls, and root biomass was 300% higher. That's the answer I could give you without knowing the physiology.
Okay. All right.
So something was happening.
Somehow figured out in your work without really understanding why, but you found a trike that plays nice with mycorrhizae.
It does.
All right.
And trichoderma is wonderful stuff. It's great stuff.
Kudos to you. Now, we're going to rip through these questions. I want short, direct answers, no stories. I want to give you enough time to be able to talk about the products. So just...
Okay, I can't hear you now because our friend came in.
Oh, man, no way. But can you hear me, Bob?
I hear you now, and I can't hear
Leighton.
That's all right. You don't need to hear me. He doesn't need to hear me.
Back off again. Bye-bye.
Okay, well, hold on, Bob. Hold on.
Yeah, hold on.
Leighton's saying you don't need to hear him.
Okay, you tell me the questions. That's fine.
Right, so Ken, instruct him. Short answer.
Question number 1.
No stories. Just blow out the answer real quick.
Okay, Leighton's saying, Bob, short answers. We want to real quick because we want to get into your products and that's part of the questions. First one from Farmer Cheryl: with respect to nematodes, what biologicals today are considered snake oil in Bob's observation? Now, I know you said you don't want to talk about other products.
Won't do it.
Okay. Cheryl, there you go.
Tell Cheryl it's the vast majority.
And Leighton said to tell Cheryl
the best way I could describe what Rhizol does is it reduces nematodes in the field to below subclinical levels. That's what we're shooting for. Okay, is that we're not temic. Okay, we are not annihilating everything in the soil, but we're taking relatively high numbers and bringing them down to the point where there's no damage to the root.
Okay, sounds good. Now, Perpetual, any way to do this yourself instead of importing pills from the USA?
What do you mean by doing it? We're going to sell internationally.
Okay, he wants to know if he can do it like Korean natural farming where they can make these amendments themselves, but obviously the processes, there's no possible way.
No, you can't. I can't do it. Okay, I mean to do what we're doing. So yeah, you can't do it individually, but trust me, a lot of people are trying. Next,
I'm sure they are. In the ProGro Bio rhizol products involved in rhizophagy and how?
Rhizophagy meaning root eating.
No.
Okay, that's what rhizophagy means. Basically, what our products will do through our enzymatic activity is it recycles roots. As roots die, as a plant is growing, as a tomato plant is growing, you get death of the root system. And as they die, they are recycled by these microbes.
So ask him this. Does it affect the rhizophagy cycle where the bacteria comes out, neutralizes and goes back into the plant and releases the nutrient?
Okay, basically saying, does it affect rhizophagy, the nutrient cycling?
Oh, it increases nutrient cycling by orders of magnitude. Yes.
No, he doesn't understand.
That's the issue. Okay, is that you're able.
Hold on, Bob. Hold on, Bob. Leighton, what?
So rhizophagy cycle is when the bacteria goes into the plant cell, loses its outer shell, releases nutrients inside the actual plant cell, and the plant cell then shoots it back out into the environment to collect more nutrient to come back inside the plant. It's called the rhizophagy cycle.
Okay, so Leighton's saying when the bacteria goes in, it loses its outer shell,
goes into the plant cell,
and then
leaves nutrients behind, pooped back out into the soil,
to
collect more nutrients to go back in.
Well, it's not only that, but the microbes die too. And what do you think they're leaving behind?
Yeah, so
they're leaving their little bodies behind, which turns into nutrients absorbed by the roots.
So let's move.
So you got both going on. It's digesting plant material and it's leaving its residues as it goes through its life cycle.
Yeah. Okay. We'll go on to the next one because I think we're not quite matching up on that one. But Squishy Mirror, I'm about to use Rootwise, Terraganics Consortium, and Microbe Life Photos. Plus, is that conflicting? So he wants to know basically if Rootwise and Terraganics Consortium is going to conflict.
No.
Okay. Quick and simple answer. Next one. Once the product's homogeneously sealed bag is open, do you have to use all of it right away?
Oh, great question. Okay. Basically, everything we sell. Okay. Here's an example of the pills that we're selling for cannabis. Okay. Zenra. Okay. These all come in resealable Mylar bags. This is a resealable Mylar bag. Okay. So the answer is if you're not using the whole bag, okay, if I'm only using a quarter of this bag because I want to treat 10 acres, I take out a quarter of the material, I use it, I zip the bag closed, and so long as I get no water in that bag, real important data now, I have kept these opened bags, meaning that they were initially opened and resealed, frozen, refrigerator, and room temperature for 3.5 years. There is no deterioration in the product. Okay? Cool. You don't want water getting into it. So long as you seal that out, You can reuse this ad nauseum.
Cool. Does it function differently in various bioregions?
Great question. Okay. And let's go back to the original things I was talking about with Leighton. We wanted to create a product in which our microbes came from such a broad diversity of bioregions and ecosystems that it is going to work everywhere. As I said, Northeast, North Central, West Coast, Arizona, Florida, Louisiana, North Carolina, Missouri, Illinois. Picture that in your head. We have used the same treatment everywhere on 43 different crops and gotten the same response. So the answer is it doesn't matter what the bioregion is.
Okay, cool. This one. Whoa, so can one add rhizol soil in compost we make to boost?
Oh, yeah. Great question. The answer is, yeah, if you got a good compost pile, go put a 5-gallon bucket with some pills or some powder of rhizol in it. Throw it on that stuff, and you are going to turn it into the finest damn mulch and humus that you've ever seen. Absolutely fantastic.
And then our last question, and it's basically they want to know what the difference is between the products. So maybe we'll go to the product page.
Let's go to the products. Yeah.
And
Okay.
yeah,
One last question. Does it?
All right. So, yeah, basically.
Hold on, Bob. Leighton has one more question.
Does it speed up decomposition in immature compost piles?
Does it speed up decomposition in immature compost piles?
Yes.
By how much?
Sure does.
By how much?
It takes me about.
I have not run the numbers on it, but I would say anywhere between 25 and 50%.
Thank you.
You can actually tell the heat coming off of it. Okay, it starts digesting fast.
Okay, let's add your stuff.
Okay, what I want to do is put up just some of the things here. Okay, our flagship product is Rhizol. This is a bag, little tiny bag that has the Rhizol in it. This is a 20-acre bag. Okay, that is good for 20 acres. We have products for virtually every major crop that is out there. And again, we take it all the way down to a tiny bag. Okay, not everybody needs a 20 or 50 acre bag. Look at this guy. It's 1/8 of an inch thick. Again, and you pour this amount of material into your watering can, your tank, your mixer, and everything. And you could fully treat a whole yard, literally, with this bag. Okay, your garden, your trees, your flowers, all from a small bag. The direction that we're moving, and
hey,
I
already. Bob,
yeah,
hold on. Leighton's got another question.
Okay.
Was that a 1 acre bag?
Is that a 1 acre bag?
Yeah, 1 acre or less.
1 acre or less. Okay.
1 acre. And by the way, very important. No one's asked this. Can you OD? The answer is no. Okay. I could take a whole bag, put it in a gallon of water, and dump it on a bunch of plants. I just wasted a lot of Rhizol. Okay. So that's about it. You can't OD, which is important. Again, okay. We already talked about talc as a way of delivering it. The new kid on the block, okay, are the two products that we are now featuring. Right now, we're using it strictly for cannabis. Okay, next year it will probably be put in a way that it's going to be used universally. And again, talk about packaging. So we created our powder, okay? And we said, how can we use it even more efficiently? And bingo, pills.
Yeah.
We knocked out pills. Notice these are brown.
Mhm.
These are rhizol pills that contain fulvic and humic acid. Is fulvic and humic acid good for microbes?
Yep.
Oh, yeah. Oh, yeah. It's an incredibly concentrated source of carbon. Then we created Zenra pills, used again in our cannabis sales, that are white without fulvic and humic. And these are used for people using over the top foliar sprays in order to get it onto the plant and dripping down into the soil, into the plant itself. So, this again, who came up with this idea? Our engineer. Okay, again, efficiency. How do you take something, by the way, this pill, one of them that you're looking at right now, that's the Zenra pill. One of those pills in a 5-gallon bucket of water, and you can fully treat anything you want with it. Hydro, Coca-Cola, okay, drip, sprayer, foliar, makes no difference. Basically, one pill for 5 gallons of water, and you've got a universal inoculant. And what's so cool about it is, remember, we talked about the fact that once you rehydrate these microbes, they go right back. Bingo,
Mhm.
they start multiplying right away. This gives it a kick. Okay, this is like giving it a shot of of stuff you get from Colombia. Okay.
Uh-huh. Now, can
Uh-huh.
this be used in compost teas? Would it actually be advantageous to add it into a compost
Sure.
tea?
Sure. 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 No, it'll make the compost tea, frankly, ain't going to do much. Okay. By putting this into it, you're multiplying the factor of efficiency of the product. So with Zenra, again, okay, whether we're talking about, we're selling them in a small bag. I showed you a picture of it. Let me see if I can get that again.
Yeah. So it
Okay.
doesn't matter if it's an aerated compost tea, anything like that.
No, not at all. Okay.
Okay.
But that little packet right there is going to contain 3 pills. Okay. And that will be sold. It's soon going to be on Amazon and other retail places. Okay. So again, who are we shooting for there? A home grower, a small grower growing 10 plants or whatever. Okay. On the other hand, if you're a California grower and you've got 80 acres, okay, of cannabis, okay, we will send you obviously at a wholesale rate, okay, enough pills to put in enough water, aka a 500-gallon tank or a tank drip irrigator or a pivot to be able to shoot it across the whole field and inoculate all of your plants. Question.
Now, international shipping, where can you ship to and where can you not ship to at this point? And where do people get it? Do they go to your
Okay,
website?
this is a good question, and that is still being worked on. Okay, there are different laws, seriously different laws pertaining to Central America, South America, Europe, India, okay, places like that. Okay, I found out that, for example, in India, where they have a lot of these biological products, there's no problem being able to ship there. Europe, you've got to jump through some hoops. Canada, you've got to jump through those hoops. So that's one of our major, I won't call it an obstacle, but challenges right now, to be able to get enough funding for the company to be able to really get the lawyers and the people involved with this up to speed in terms of the international shipment. On the other hand, I'm going to say something, and I'll probably get in trouble with this, but I'll say it anyway. Let's assume somebody in Germany said, wow, this is the best thing since sliced bread. And we go, yeah. Do you know how much it would cost them licensing from us? to build an entire manufacturing facility in Germany to be able to provide Europe with product, the answer would astound you. Okay. It would astound you. Okay. It is not that much. What it comes down to here is, other than the lyophilizer, okay, which is, as we said, you're talking about a $500,000 machine, but to a huge company, that's not a lot of money. The rest of it is basic microbiology, knowing what to do. That's the IP is the value. Okay. Anybody can grow bugs in a tank. How you grow it, the nutrients you use to grow it, how you process it could be done. So we're going to have interest in that too. Is that,
Cool.
we thought, I told you, we try to think about everything. Do we want to simply produce 100% of this material in the United States? It's a possibility. On the other hand, can you license the technology to different countries or different continents for them to produce it themselves? The answer, again, is yes. Frankly, boy, would I love to see somebody manufacturing this in Africa. They
How
need
about,
this
okay,
bad.
hold on. Yes, they do. Leighton, you had a question?
It's
Yes.
a point of interest.
Point of interest.
He can sell that product to the cannabis industry, but he cannot sell it to the agricultural industry in California.
Okay, he's saying you can sell it to the cannabis industry, but not the agricultural industry in California.
Interesting. I'm glad to know that. Yeah.
Well,
Australia. We have a lot of people from Australia. Do you ship there into
We
Australia?
we can ship to Australia without a problem. I've checked that out.
Okay.
Yes.
So
Now, where do they go? Where do they go to actually, buy it? Is it your website?
It's okay. The website is still being built. It's being massaged to get all this information on it. I would say hang on for a matter of weeks and
Okay.
you'll be able to get it. Also, on the website, there is a place to email or text, okay, to our people that are taking care of these questions, the marketing people. So if they have any questions, just go to our websites. Okay, the website for the cannabis is Canopy Bio Labs. Canopy, as in a plant canopy, biolabs. And the questions for all the rest of agricultural crops, it's ProGro Bio.
So, Ken,
And you'll find it. It's on Google. Yeah.
So in California, you would have to go through what's called CDFA, and they would require that you gave a coliform unit of each and every microbe that you put in there, which would kind of ruin your IP. Have you looked into that in California?
Okay, basically, he's saying it's the same thing in Canada where they wanted your complete list for California.
This is the problem. One of the things that we were very careful about, and I was very careful about, I'm the scientist dealing with this stuff, is making sure, okay, I mean, there are liquid products out there that will remain nameless that have a huge following right now in the United States. They have pathogens in the liquid. They admit they have pathogens in the liquid. Okay, and I'm not only talking about plant pathogens, I'm talking about animal, human, and fish pathogens. Okay, and how they get away with that, I just don't know. Obviously, they're not OMRI labeled, they're not EPA labeled. They're just juice, again, the snake oil that they're selling. We make sure that we absolutely are dead set on making sure that our microbes are safe. As a matter of fact, Europe, okay, good, it's actually a good question. European Union requires fish toxicity studies. Okay, good idea. Good idea. Okay, you don't want this stuff running off from your field into the stream and killing fish. So what did I do? I bought a tank, I bought fish, and I started doing studies. Okay, by putting 1x, 10x, 100x, 1000x the concentration as administered by the EU for toxicity, even at the
Mhm.
highest level, no toxicity. So, it's a matter of jumping through hoops. It's a matter of getting the right contacts, quite frankly, to be able to show them that you've got a safe product. And, by disrupting the ability to export it, all you're doing is not permitting farmers to have something that could be a game changer for their operations, particularly in Europe, where they're getting very tight on pesticides, getting very tight on GMOs. So, these are challenges that are there. And of course, let's talk about the major challenge. Okay, basically, the products I just showed you are equivalent to a bag of carcinogenic furidan polycyclic aromatic phosphorus chemicals that will kill you. That's the same way the EPA looks at a bag of pills. Okay? No difference. No difference. You have to pass the same thing, which, as I said, costs a minimum of $500,000 of independent trials. Okay, toxicology, everything else, to show that what you've got is safe. On the other hand, what's so funny about it is if I don't call this a biopesticide, this is no longer a biopesticide, it's a soil amendment.
Right.
I could put this on every plant and every tree growing in the United States legally. Go figure that one. It's archaic, it's anachronistic. And by the
Yeah.
way, the EPA knows it. I've gone up there. Okay. I was on K Street. I visited the people. I said, what in the hell's going through your guys' heads? And they say, Dr. Bruck, we don't make the laws, we enforce them. Okay. And I know that there's outreach now to senators and other people because biologicals, particularly when the big boy companies start putting out their products, you're going to see an ipso facto turnaround on the spot because these laws are ridiculous. And of course, for a small company, by the way, I failed to mention, it's not $500,000 for this pill. Each organism, 40 of them, are $500,000 each.
Yeah.
Each, okay, in order to get clearance to get an EPA registration as a biopesticide. So, these are challenges that we've got, but we're sticking to our guns.
So, what is the cost for these units?
What is the cost of these units? Like, how much is a bag?
Okay, it's different for all things. Okay, it's different in the volumes that you buy. Again, if you go on our pages right now, you'll get some of those numbers.
Gotcha.
Okay, I mean, if you're buying a 3-pill bag per unit, obviously it's going to be different from if you're buying 1,000 pills. So it really depends on what you're looking for.
Okay, now the last question I had,
Yeah,
does it save time with breakdown of animal manures?
great question. Okay, the answer would have to be yes, even though I've not done animal manure. Basically, remember the 3 words, okay? Carbon, lots of animal manure is carbon. Moisture, you got to have liquid in order for them to grow, and a reasonable temperature for them to be able to multiply and stay alive. But actually, the degradation of manure, okay, it's funny you say that. What are we famous for in North Carolina besides tobacco? What's our number one commodity?
I'm from
Oink,
Canada, so I have no
oink,
idea.
oink, oink,
Pigs,
pigs.
okay.
Pig poop goes in lagoons. Okay. It's shoveled right out of the houses by the ton into these sludge lagoons that you can smell from 20 miles away. Okay. I have tried on several occasions to get permission or a license to apply our microbes to a hog lagoon to see what happens. Obviously, one treated, one control. Hasn't happened yet, but I'll bet my bottom dollar that we could more quickly remediate the organic matter in those lagoons. And I'll even go further by saying pathogens, okay, worms, nematodes. We could probably have a tremendous remediation effect. Soon to be done, thought of numerous times.
Have you done cattle lagoons?
I have not. I've not.
Okay.
Yeah, up in
There's
Winnipeg,
another
you ain't got no pigs. I got it.
Well, we have some, but not like what you guys have down there. But
Okay.
yeah, huge cattle, and there's
Oh,
lots
yeah.
of in the States that, you know, dairy farms especially, where you could probably get permission to do that, because I know a lot of people are
I
doing
know.
things with their dairy lagoons.
We've got to get the staff to be able to do it. Another thing that's very important is there's more and more work being done right now on water remediation with microbes. Okay, right now the big issue in the United States is PFAS and PFOS, the
Yeah.
forever chemicals. Okay, these are cyclic hydrocarbons, which again is a carbon source in which microbes can break down these forever chemicals into lower class of chemicals, which could be much more easily remediated. So there's loads of research. I mean, you know, again, but back to what Leighton said, you know, the world is filled with microbes. We've hardly touched the surface on being able to fully understand what these things can do, but we need to do it because we're killing the world with chemicals right now.
Yeah. How about biosolids? Have you done anything with biosolids?
Great question. Okay, I tried to get, we have some huge municipal water treatment plants here with biosolids. We've talked several times. I actually have one of my ex-students is the manager of the entire municipal treatment operation in Wilmington, North Carolina. And we've tried to get started on looking at remediation of biosolids. Great question. Again, I'll bet my bottom dollar that we can make progress on that. We have not done it yet. Great question.
Okay. We're only have a couple of minutes left, so I'm gonna say we're gonna cut it here. Poor Leighton, he can't say much,
Well,
but I know we want
I
to
can
thank
say
you.
it through you. So
Well, thank
tell
you,
him-
Leighton. You're a brilliant man. You've got bright questions. I hope we connect again to continue this story.
Thank you. I was going to say, tell him, please. Thank you so much for giving us the 2 hours, and I definitely want him back on in 3 to 6.
And if you give it to that cute puppy, I don't know what it'll do.
But he's
That's
saying,
Cheeto.
that's Cheeto and Bob. He's saying we're gonna have you come back on. There's, you know, you've done an amazing job, and we want to know more and more about your research, etc. Thank you so much, and you know, I'll definitely make sure that I connect you to again with the emails, because I
Great.
know behind the scenes. Oh my God, I'd love to be a fly on the wall when you guys are yapping behind the scenes.
Well, it's been a pleasure. I really enjoy you guys a lot. Thank you for inviting me. And the story goes on. We have a lot more to do.
Yeah, we do, and a lot more to talk about. So
Okay, thank you guys.
Okay, guys, we're gonna end the stream. Talk to
Okay.
you soon. Bye-bye.
Thank you. Bye-bye.